Welcome to PRISM Rounds, the audio journal club where we break down one study at a time for ICU, ED, and pulmonary/critical care teams, physicians, trainees, nurses, pharmacists, and respiratory therapists. A quick note before we start, this episode is created using AI-generated voices. I originally made these episodes to support my own learning and review, and I'm sharing them publicly in case they're helpful to other busy clinicians and learners. Because the voices are synthetic, you may occasionally hear mispronunciations of medical terms, drug names, or author names. Thanks in advance for your patience. Everything you'll hear is for education only and not medical advice. Let's get into today's paper. This week, we discuss a familiar ICU dilemma. The blood culture shows gram-negative rods, and everyone wants to know whether we are using the right antibiotic. The FAST trial, published in JAMA, April 2026, tested whether rapid susceptibility testing could move those decisions faster. It did, but without a clear improvement in 30-day outcomes. In my opinion, the study still matters because faster results may be most useful when resistance is high, empiric therapy is wrong, and stewardship teams can act quickly.
Welcome to PRISM Rounds, Season 1, Episode 39. Let's get right into today's deep dive. Imagine you are getting that dreaded 2 a.m. bedside call.
Oh, man. I know that call all too well.
Right. You are the clinician covering the intensive care unit, the pager goes off, and the message is incredibly tense. Your patient's blood culture is just flagged positive for gram-negative rods.
And of course, the patient's getting hypotensive.
Exactly. Their blood pressure is just dropping, and the bedside nurses are urgently asking you what antibiotic you want to use. And in that moment, you are effectively flying blind.
Yeah, totally blind.
You have to decide, do I keep them on their current broad-spectrum empiric therapy? Do I escalate to something even stronger? Or do I try to narrow it down?
And you're making this call days before the final laboratory susceptibility data will actually tell you exactly what bug is swimming in their blood.
Right, and what specific drugs will actually kill it.
It is arguably one of the most high-stakes, stressful decisions in hospital medicine. I mean, historically, you were waiting up to 3 or 4 days for that final report.
Which feels like an eternity in the ICU.
It does. And because the stakes of getting it wrong in septic shock are absolute, clinicians tend to err heavily on the side of caution. We blast the patient with the broadest possible spectrum of antibiotics.
Yeah, you hope you hit the target, but you also know you are kind of carpet bombing their microbiome.
Exactly. Potentially trashing their kidneys and, creating collateral damage that might haunt them later.
Which brings us to the core mission of today's deep dive. We are looking at a crucial question for anyone working in or studying healthcare.
Does getting that antimicrobial susceptibility data hours, or even a full day earlier, actually save lives?
Or is the reality of treating severe infections far more complicated than simply speeding up the lab results?
Spoiler alert, it's usually more complicated.
Always. So we are pulling our answers from 2 primary sources today. First, the FAST randomized clinical trial.
And second, its accompanying editorial written by Dr. Arjun Srinivasan.
Right, and both of these were published in JAMA in April 2026.
So to understand how researchers attempted to solve this 2A dilemma for you, the listener, we need to look at how they stress-tested rapid diagnostic technology on a massive global scale.
Yeah, tell us about the trial setup.
Well, the FAST trial, which stands for Fast Antimicrobial Susceptibility Testing, was this incredibly ambitious open-label trial. They enrolled 850 hospitalized patients.
Wow, that's a huge cohort for an ICU trial.
Yeah. And this included both adults and children, all of whom had confirmed gram-negative bloodstream infections.
And crucially, they didn't just run this in, one comfortable, highly resourced academic center in the U.S.
No, not at all. They operated across 7 different centers in Greece, India, Israel, and Spain.
Right. They intentionally went into environments with a high prevalence of antimicrobial resistance. They went to where the bugs are the absolute toughest.
Exactly. But before we get into the results, we really need to clarify the mechanics of the test itself.
Yeah, the sources note they compared standard testing against a rapid phenotypic test, specifically using a system called Vitek Reveal.
Now, the term phenotypic is doing a lot of heavy lifting here.
Right, because under standard care, when that blood culture bottle beeps positive, the lab has to take a sample, smear it onto an agar subculture plate, and then wait.
Wait for days, yeah.
Yeah. Wait days for the bacteria to physically grow into visible colonies before they can test different antibiotics on them. It is essentially a multi-day science experiment.
But this rapid phenotypic test, it skips the Petri dish entirely.
Oh, really? How does it work?
The machine looks directly at the soup from the positive blood culture bottle. It analyzes how the bacteria behave in the presence of drugs in real time.
That is a profound technological leap.
It's massive.
Yeah.
So one group of patients in the trial got this direct-from-bottle rapid test alongside standard care, while the control group received standard care alone.
But there is also one more vital variable the researchers controlled for, right?
Yes, and this is key. In both arms of the trial, every single patient had their case reviewed by an antimicrobial stewardship team.
Ah, okay. So regardless of how fast the lab results came back, an expert pharmacist or infectious disease clinician was reviewing the data and recommending the best drug.
Exactly. It leveled the playing field clinically.
So with that playing field leveled, the researchers needed a way to measure if the rapid test actually worked.
And they didn't just look at a binary, did the patient live or die metric?
Right. They used a patient-centered measurement called the DOOR outcome. DOOR stands for Desirability of Outcome Ranking.
I actually love when trials use this.
Same. It is an ordinal outcome measured at 30 days. So the best category is being alive without any deleterious events.
And then the middle tier is being alive but suffering from deleterious events.
Right. Things like acute kidney injury from toxic antibiotics or catching a secondary hospital-acquired superbug like C. diff.
And of course, the worst outcome is mortality.
It adds such necessary nuance to critical care research. Surviving an infection but ending up on lifelong dialysis because of the drugs we use to save you is vastly different than surviving with your organ function completely intact.
Completely different. So the trial was structured with the DOOR outcome as the primary finish line.
Okay, so what was the bottom line?
Well, the headline finding from the trial was that the rapid testing did not meet superiority.
Oh, really?
Yeah. The probability of a better DOOR outcome with a rapid test was only 48.8%.
So less than half.
Right, with a 95% confidence interval ranging from 45.3 to 52.4%. Statistically, it failed to prove that it was definitively better than standard slower testing.
Hold on. That defies clinical logic.
I know, it really seems counterintuitive at first.
Right. If we have this highly advanced machine that skips the agar plate, giving us the exact identity of the bug and telling us exactly how to kill it a full day earlier, why didn't outcomes massively improve?
That's a great question.
It is like getting the answers to a test early, but still failing because you studied the wrong material. What went wrong in the clinical reality of these hospitals?
Well, your analogy is spot on. Having the answer key early only provides a life-saving mortality benefit if 3 very specific conditions are perfectly met at the bedside.
Okay, lay them out for us.
First, the patient must not already be on the right drug.
Makes sense.
Second, the patient has to be sick enough that a difference of a few hours actually dictates their survival.
Okay, and the third?
And third, the correct drug identified by the rapid test has to actually be sitting in the hospital pharmacy ready to be infused into the patient's arm.
Logistics. Let's examine that first condition, not being on the right drug.
So the trial data shows that over 60% of the enrolled patients were already receiving effective empiric antibiotics when they entered the study.
Wow, 60%. So going back to our 2 a.m. scenario, the clinician got the call, evaluated the hypertensive patient, and made an educated guess.
Right. They started a broad-spectrum antibiotic, and it just happened to be the correct one to kill that specific gram-negative rod.
So when the rapid test comes back 14 hours earlier and informs the clinical team that their empiric choice was correct, I mean, it provides immense psychological relief.
Oh, absolutely. You can finally breathe, but it doesn't alter the patient's immediate trajectory at all.
Right. They were already receiving the correct molecules circulating in their bloodstream.
Exactly. This single factor wipes out the potential mortality benefit for nearly 2/3 of the trial participants right out of the gate.
That makes total sense. And regarding the second condition, being sick enough that hours matter, the sources show that only about 1/4 of the participants actually presented with severe septic shock.
Yeah, so if a patient has a milder form of bacteremia or Conversely, if their infection has already triggered irreversible multi-organ failure before they even enrolled in the trial, optimizing their therapy a few hours earlier simply isn't going to dramatically shift their 30-day door outcome.
The margins of survival in sepsis are highly dependent on illness severity.
But the third condition is perhaps the most revealing and honestly the most frustrating aspect of the entire FAST trial.
The pharmacy issue.
Yes. Over half of the participants were enrolled in regions, specifically the centers in India and Greece, where newer first-line therapies for highly resistant organisms simply are not accessible.
We are talking about modern drugs like sulbactam, durlobactam, which are considered the gold standard in highly resourced nations.
Right, but those were effectively off the table for these clinicians.
It is like being handed the exact blueprint to defuse a ticking bomb 14 hours early, but when you open your toolbox, the wire cutters are missing.
That is exactly what it felt like. The sources even highlight a specific logistical nightmare. During the study, Greece experienced a national shortage of Astrionam.
Wait, a national shortage right in the middle of the trial?
Yes. And Astrionam is a highly specific antibiotic relied upon to treat some of the most stubborn carbapenemase-producing bacteria.
So the diagnostic machine performs perfectly. It analyzes the bacteria, it identifies the vulnerability, and the report tells the clinician to use Astrionam.
But then the clinician calls the pharmacy only to be told the drug does not exist in the country.
That is just heartbreaking. It forces clinicians into a devastating corner.
It really does. The trial data reveals that 10 to 15% of patients had to be treated with older, notoriously inferior regimens. They had to fall back on drugs like colistin.
Ugh, colistin. That is a drug most clinicians desperately want to avoid.
Oh, totally.
It is an older agent, it is highly nephrotoxic, meaning it just destroys kidney function, and it is generally less effective than modern alternatives.
So when you are diagnosing a pathogen at light speed but are forced to treat it with highly toxic tools developed decades ago, patient outcomes cannot improve.
The trial exposes a massive vulnerability in our global healthcare system. Diagnostic technology has outpaced the pharmaceutical supply chain.
Exactly.
But, if the rapid test couldn't help the majority who were already on the right drug, and it couldn't help the patients where the right drug was out of stock, we have to look for the signal in the noise.
Because the trial wasn't a total failure.
Right. The sources point to massive hidden wins in the subgroup data, specifically regarding patients battling the toughest bugs.
Yeah, let's talk about those. 44% of the trial patients had organisms resistant to cephalosporins or carbapenems.
For the listener, these are the nightmare bacteria. These are the bugs that have evolved to eat our strongest absolute last-line broad-spectrum antibiotics for breakfast.
Right. In the general trial population, about 60% of patients were likely on the right empiric drug.
Yeah.
But when we isolate the carbapenem-resistant infections, that number plummets.
It just plummets.
Only about 38% of these patients were receiving effective therapy at baseline.
Wow. Because standard empiric drugs do nothing against these highly mutated superbugs.
Exactly. So these patients were effectively receiving zero beneficial treatment while their infection just raged on.
And this is where the speed of the Vitek Reveal system becomes a literal lifesaver.
Yes. For patients with carbapenem-resistant infections, the time to getting on effective therapy was 9.5 hours with the rapid test.
And with the standard agar plate test,
It took 28 hours.
while the sources note the confidence interval for this specific time difference did cross zero, mostly due to sample sizes in the subgroup, the clinical reality of that gap is staggering.
An 18-hour head start on a resistant superbug is massive.
When a house is burning down, 18 hours is the difference between replacing some drywall and watching the entire foundation collapse.
That's a great way to put it. And the clinical impact of that speed is undeniable.
What did they see in terms of outcomes for this group?
Because these specific patients were moved onto effective therapy so much faster, the proportion of them who remained hospitalized at 30 days was roughly 13% lower in the rapid testing group.
Compared to the standard testing group, that's huge. Keeping a vulnerable patient out of the intensive care unit for extra days translates to a massive reduction in hospital-acquired complications.
Definitely. And zooming out from just the carbapenem-resistant group, the rapid test shortened the overall time to antibiotic escalation or de-escalation by about 14 hours across the board.
Right. The median time to make a definitive modification to any patient's antibiotic regimen was 14 hours faster.
That is 14 fewer hours of uncertainty for the clinical team and 14 fewer hours of unnecessary broad-spectrum exposure for the patient.
Which brings us to the interpretation conundrum. If the primary door outcome, the main measure of success, was technically negative, how should a hospital system view this multi-million-dollar diagnostic technology?
Yeah, it's a tough pitch to administration, but Dr. Srinivasan's editorial tackles this directly.
What's his take on it?
His core argument is that while FASTER did not cleanly win the primary mortality endpoint in this specific trial, FASTER information is an absolute non-negotiable necessity for the future of antimicrobial stewardship.
Right, because the trial uncovered a fascinating behavioral quirk among the doctors. The researchers noted that stewardship recommendations and the clinicians' actual acceptance of those recommendations were significantly higher in the rapid testing group.
The psychology here is incredibly important. Imagine you are a doctor who just admitted a very sick patient. You put them on a massive dose of broad-spectrum antibiotics.
And then 3 days go by and the patient finally stabilizes.
Right. If the lab finally calls on day 3 and says you can narrow the drugs to something weaker, human nature makes you hesitant.
Of course. The patient is surviving. You don't want to rock the boat.
But if you get that exact same data just a few hours after admission, before you have settled into a clinical routine and before the patient has been stable for days, you have the confidence to immediately de-escalate.
The intervention provides clinical confidence.
Exactly. Dr. Srinivasan points out that rapid AST should not be sold to hospital administrators purely as a magic bullet for mortality. Setting that expectation misunderstands the complexities of sepsis.
Instead, it must be viewed as a workflow intervention.
Yes. The technology alone does not save the life. It requires the speed of the lab, combined with the vigilance of the stewardship pharmacist, combined with the bedside clinician actually acting on the data to change the prescription.
And when that workflow operates efficiently, faster de-escalation prevents prolonged hospitalizations. It prevents the acute kidney injury caused by days of toxic empiric drugs.
And on a systemic level, it stops the creation of more superbugs.
Right. The longer you carpet bomb a patient's microbiome with broad-spectrum antibiotics, the more you encourage the surviving bacteria in their gut or on their skin to mutate and develop resistance.
Every single hour of unnecessary broad-spectrum coverage is an evolutionary opportunity for resistance to develop.
So by cutting 14 hours off that exposure time across hundreds of patients, rapid AST is protecting not just the patient currently in the bed, but the next 10 patients who will eventually be admitted to that same hospital wing.
It's a public health win disguised as a negative trial.
So bringing this all home for you, the listener, the FAST trial, despite its technical failure to meet statistical superiority on the door outcome, is an incredibly revealing look at modern medicine.
It proves that rapid diagnostic testing radically improves the timing of our antibiotic decisions.
It gets the fire department to the worst, most resistant fires 18 hours faster. But the true lesson here is that the last mile of medicine is entirely about implementation.
The last mile requires drug availability, accurate patient selection, and a clinical team ready to act on the data.
Right. The rapid test is a highly advanced compass, but the healthcare system still needs a functioning vehicle and clear roads to actually reach the destination.
Well said.
Which leaves us with a lingering, somewhat unsettling thought. Diagnostic technology is becoming increasingly rapid.
Oh, it's moving incredibly fast.
We are moving toward a near future where we might be able to predict bacterial resistance instantly, perhaps using AI and genetic sequencing right at the bedside the moment a patient arrives.
Definitely coming.
But if we achieve that perfect diagnostic clarity, are we heading toward a future where we know exactly what is killing a patient in real time, but remain entirely powerless to stop it simply because physical, pharmaceutical supply chains and drug shortages fail us?
Diagnosing the fire instantly offers little comfort if there is no water in the hose.
Something to mull over the next time you are staring at a blood culture report at 2 a.m.
Thank you for spending your time with PRISM Rounds. If this episode helped you think through the paper or offered something practical for the bedside, I'd be grateful if you'd like, follow/subscribe, and share it with a colleague. It's the simplest way to help this resource reach other busy clinicians and learners. I'm also trying to make the podcast better with every episode. Please leave a comment with what you found most useful, what felt unclear or too long, and what topics or papers you'd like covered next. If there's anything I can do to improve, pacing, structure, level of detail, or audio quality, I genuinely welcome the feedback. Thanks again for listening, and take care. Educational use only. This is not medical advice.